Okay, so good afternoon, everybody. And again, it's a pleasure for me to have Dr. Jyoti Lakshmi Vadasuri here today with us on the third day of the DST-STUTI workshop. And she's a brilliant scientist and a good friend. So it's a pleasure to introduce her to all of you. So Dr. Jyoti Lakshmi, I'll just give a brief introduction about her and then we'll go on to her talk. So she did her MSc from Indian Agricultural Research Institute, Delhi, and then her PhD from Germany.
Jenny,
yes.
You didn't let me welcome her first.
Oh, but okay. Now you have already welcomed. So do you want to?
Yeah, I want to officially welcome her.
Okay, please go ahead.
Because we wanted her for so long, and it's unfortunate she's not able to come, but nonetheless, she's participating. And
please go ahead, sir.
Am I audible?
Yes.
Yeah. So these participants will always think that any expert or speaker coming during the workshop, they are very friendly to all of us. But what can we do? We are all friends. So, and like Jenny was about to introduce, she's very good. And I wish if you could visit the facility also, but probably not possible this time. But we'll have a link so you could visit them again and see the facility yourself. But she will tell you the application part during next 1 hour. Ujjani, please go ahead.
Okay, thank you, sir. So she did her MSc from Indian Agricultural Research Institute, Delhi, and then her PhD from Germany, from Friedrich Schiller University. She was a postdoctoral associate at Cornell University, USA, and then followed by this Max Planck Institute for Chemical Ecology, Germany. And needless to say, she has a bag full of achievements and awards. She is the EMBO Global Investigator, and that just happened very recently. Then she was the head of Max Planck India Partner Group in Chemical Ecology. She also got the Wilhelm Pfeffer Stiftung Prize, German Botanical Society, awarded for the best paper in 2010, most probably while you were doing your PhD, Jyoti.
Yeah.
So, and if I am pronouncing the German names not right, please excuse me. And she also got the Max Planck Society Postdoctoral Fellowship and the DAD Postdoctoral Fellowship. So in all these past years of PhD, postdoc, and now she's the PI at NIPGR. So she's the staff scientist 5 at NIPGR. That is in Delhi. And she has ample publications, including very prestigious journals like Plant Cell, Plant Physiology, Plant Journal, New Phytologist, PLOS Pathogens, so a whole lot of them. And she is interested in the molecular mechanisms by which plants defend themselves against a wide array of insect herbivores, and uses this knowledge in developing insect control strategies. So she uses forward and reverse genetics approaches, biochemical techniques, metabolomics, and imaging to bring about, novel discoveries in plant defense mechanisms. And her group is actively involved in doing this. So today she's going to tell us about the use of high-resolution metabolomics for plant improvement. So without any more delay, Jyoti, it's over to you.
Thank you very much, Jenny. I will just share my screen and start the presentation. Yeah, I can see that the slide is visible.
Yeah.
So thank you very much for the invitation. And I'm really sorry to all the students, especially that I couldn't be present in person because of ill health. But next time there is any event there, I hope I can come in person. Nevertheless, today's talk is on using metabolomics. How can the applications of metabolomics, how we can use it for crop improvement. I will be explaining 2 ways which I have used it in my lab to enhance insect resistance in one case, and in another case where we have worked out the mechanism in plant growth promotion using metabolomics. Yes. So my lab, essentially, the main focus of the lab is on plant-insect interaction. As you know, that plants are attacked by an array of insects, which can attack, almost all parts of the plant: leaf chewing larvae, phloem sucking aphids. They attack fruits and roots everywhere. And we are trying to understand how do plants resist against these herbivores? What are the metabolites involved in this process? This is the field of chemical ecology, which tells what are those metabolites which mediate the interaction between plants and insects. So the insect herbivore we are studying is Podoptera, which is a generalist insect pest. It's called the common cutworm, and you can see it probably in your garden as well. It attacks many parts of the plant leaf. A subspecies of it, Spodoptera frugiperda, is a major pest of maize, which has migrated to India from US owing to the climate change very recently. So there is a renewed interest on research on how plant defends against Spodoptera. Why I'm telling about maize is because the work that I'll be presenting today will be more on maize. So we are also doing a lot of basic research trying to understand the genes which are involved in this resistance pathway. The second topic which is ongoing in my lab is a continuation of my PhD work. So I've actually worked on a very different topic for my PhD, which is on a fungus called Piriformospora indica, which is a endophytic fungus on the roots of plants. So what this fungus does is that it promotes growth of plants, immaterial of what kind of plant. Every plant tested so far has promoted growth. You can see here, this is Arabidopsis without the fungus, and this is, you can see a bigger Arabidopsis, it promotes growth. Also tobacco, various systems, wherever Chinese cabbage here, it can colonize roots, only roots of wide array of higher plants, and it provides nutrient uptake, enhanced disease resistance, apart from growth promotion response. So these are the two systems or models which we are studying, and we are using various techniques to study what is the mechanism in each of this plant biotic interaction. We'll start with the herbivory part. As I told you, my lab essentially works more on insect herbivores and what are the metabolites involved in the process. So when we talk about herbivory, this is a picture of an insect eating on a plant. And you can see that the plant recognizes both the wounding and the oral secretion of the insect. And it is known that it activates a very fast phytohormone pathway called jasmonate signaling. This is known from a long time that jasmonates are activated. And this jasmonic acid pathway in turn leads to production of secondary metabolites, which can act as chemical defenses. And also there can be trichomes, which are physical defenses and defensive proteins, etc. Now, all this makes the plant less tastier to the insect, or it makes the digestion of the plant tissue difficult for the insect. So in this way, it provides a direct defense strategy. Now, there is also an indirect defense strategy whereby the plants release volatiles into the air, and which in turn attracts predators or parasitoids or insects which will eat on the pest. Okay, so then this gives it a kind of indirect defense in the way that it calls help for other insects to eat it. Now, in this whole strategy, as you can understand, direct defenses in plant-insect interaction are mediated by secondary metabolites majorly. And also upstream to the secondary metabolites are plant hormones, which if you have seen in the previous slide, it is the jasmonic acid signaling which leads to the activation of direct defenses. So what are plant hormones, and what do we know about it? We know that plant hormones regulate almost all stages of plant life cycle. For example, embryogenesis, germination, flower development. They also provide a biotic and biotic stress tolerance, for example, against herbivory. So a lot of plant hormones and their role in various plant processes are known. Now, when you talk about plant hormone, the major defense hormone, those hormones we associate with plant defenses are jasmonic acid and salicylic acid. When you talk about drought, it is abscisic acid. And then there are also growth hormones like cytokinins, auxins, gibberellins, etc., which regulate many growth processes. So in this way, they regulate many plant mediated responses. Now, since we are interested in plant defense pathway, that is how plants defend against insects. For us, the major pathway is this jasmonic acid, the hormone, its bioactive form, the jasmonic acid isoleucine, salicylic acid, ABA, cis-OPDA is the precursor of jasmonic acid. So we would like to measure the levels of all of this when herbivory is happening. So as I told before, jasmonic acid is the key pathway which controls 90% of defenses against all lepidopteran pests, whereas in plant microbe interaction, majorly they are salicylate. And we use JA, it's bioactive JA, ILA, and OPDA as markers for herbivory defense in plants. Now, what will happen if jasmonic acid pathway is down-regulated, or if it's not there? Why I'm telling is to show you that it is so critical in this whole response. So these are plants in which the JA receptor, that is the protein which senses the JA phytohormone, is knocked out, or we don't have that gene here. And this is the wild type plants. They have been eaten by insects for equal amount of time. And as you can see in this plant in which there is no JA receptor, or the plant cannot sense JA, they've been completely eaten up by the larvae of the insect. So they are extremely critical for most of the responses against chewing insects. Now, when I started my lab many years ago, the major problem in measuring the phytohormone was what we faced, because plant hormones, though they are many, as I have described to you, they are extremely low in quantity in plants. So we always have to enrich them by various extract and purify, and it has been extremely difficult to analyze plant hormones or critically quantify it in plants. So there are only few labs worldwide who are doing this phytohormone quantification in a high throughput manner. So earlier, since many of the phytohormones are organic acid, GCMS was the major technology which was used after after derivatization. However, this whole method of GCMS based analysis of phytohormones was extremely time consuming, and it was not a high throughput method. So we were able to analyze SA, JA, but not all isoforms of this JA. So there was a need by which we could, make this phytohormone quantification a very high throughput method. So these are just the levels of phytohormone in different plants. So as you can see here from poplar, pisum, arabidopsis, and you can already see that their levels are extremely variable, and you can hardly see anything in jasmonic acid isoleucine. This is the most active form of jasmonic acid. So they are so low abundant. So it is extremely important that the methods that we use for quantification are also so good that we can detect it properly. So we use a triple quadruple mass spec. I will come to the data that we obtained just to give you a short overview of how we have used metabolomics to solve this problem is what I am trying to explain here. So the triple quadruple mass spec, we use a QTRAP 6500 here, and this was used over GCMS because the analysis time is very short here. We could get high selectivity, and we could analyze in raw extract. So the extraction procedure is essentially methanolic extract. We are taking methanolic extract and using isotopically labeled standards for each of the hormones. So for example, JA, if I want to quantify JA, ABA, SA, everything together, I already pre-add isotopically or deuterated standards of each of them to our sample mix. So the standards also go through the same level of sample preparation as the actual phytohormone, which is in your leaf. Now, the deuterated JA, for example, will have a different MORC value, or a retention time, which is different from your, normal JA. Imagine JA elutes at 6 minutes. So your deuterated JA will elute at 6.5. So you can, you can differentiate between your actual GA in your sample or your hormone in your sample and what you have added externally. Now, why are we adding externally this deuterated hormone? Because we have to quantify it in exact terms. I know how much deuterated JA I'm adding in each sample. So based on that, we will quantify what is in our unknown analyte. So this is usually when you do targeted metabolomics, you run a standard curve, and based on the standard curve, you quantify your metabolites. However, for plant hormones, this method is not good because plant hormones are extremely low abundant. So during the sample preparation, a lot of it is, you're losing a lot of things. So use of isotopically labeled internal standard during the sample preparation is done for plant hormones, and this makes this method highly foolproof, and you get exact quantification of hormones. Now, we are also able to quantify all the hormones in a single run. So this is, for example, SA, ABA, in a run of say 20 minutes, all the hormones we are trying, we have developed a method where which they elute at different times, and we can quantify each of them. This is using the MRM method, and this we have used for all our studies further. Now this is with the hormones. So as I told you, plant insect defense not only depends on plant hormones, but also a lot on secondary metabolites. So major metabolite classes are phenolics, nitrogen containing compounds, and terpenoids. And during various, different plants have specific metabolites. For example, poplar, mainly, in poplar salicinoids are high. In Visia, for example, lectins. When we talk about Arabidopsis, it's mainly glucosinolate and camelexin. In maize, it's mainly terpenes and benzoxinoids. So the defensive secondary metabolites in plants are also diverse, and their roles in herbivory, in many of them, have been characterized as well. Now I'll come to the first case study where I'm using metabolomics for anything related to crop improvement or on a basic translational level. So this example is of maize and a very bad insect pest of it called fall armyworm. The armyworm comes from a name because this insect comes and attacks the field of maize like an army or like in big groups, and it's a major problem, causing 73% yield loss. Now, benzoxinoids, which are the major metabolites which are known for defense in maize against this insect pest. So we started a project in collaboration with the Indian Institute of Maize Research when this insect became a big problem in India. Because there are no maize hybrids or genotypes in maize which are resistant to this specific insect. So essentially, we have screened a large germplasm of maize. We are looking for those maize plant which is resistant to this insect. Okay, they are scored based on their rating, and we have identified two genotypes, DMRE63 and CML71, which seem to be resistant to the fall armyworm in collaboration with this IIMR in Ludhiana. Now, these are the three genotypes, two genotypes which are resistant, and this is the susceptible variety. Okay, this is if you feed insect on them on a no choice assay, the insects can feed more on the susceptible variety and they feed very less on the resistant variety. So these three genotypes, we took it for identifying the metabolomic basis of their fall armyworm. Fall is that insect's name for resistance in maize. We wanted to know what gives these two plants a higher resistance than the others. As I showed you before, jasmonates are a major markers for plant defense against herbivory. So we started with the quantification of all the hormones. As I told you, they are high throughput method. So we are able to quantify not only JA and JA isoleucine, but also the catabolic products of it, OH-JALA, COH-JALA, and OH-JA as well. And we compared the levels of this hormone in the susceptible variety, the BML6, in the resistant variety, which is CML71 and DMRE63. This is in the level of these hormones in plants when there is no insect. Yeah. And after 24 hours of herbivory, you can see the same, heat map, and you can easily make out that in this resistant variety, the levels of all the hormones are highly, it's very high. Whereas in this second resistant variety, which is CML71, there is a moderate induction of the jasmonates. And now this is your susceptible variety, BML6. So in the BML6, there are some jasmonate which seem to be increased. And this is the inactive form of JA, which is called the JAOH. I'll come to it later. But mostly it is the inactive forms of JA which are upregulated in the susceptible variety. So in nutshell, the BML6, which is a susceptible genotype, shows no induction of the bioactive JA and JA-LA, or very less induction. The resistant genotype DMRE3 shows very high induction, whereas CML71 is showing a moderate induction of jasmonate on feeding. Once we got this data, we have done a 24-hour herbivory, and it is a rather late time point for measuring JA. Ideally, you should measure it at a faster time point. So we also measured it at 3-6 hours after insect feeding. So now these are the levels of JA and JA isoleucine using the method I just described to you, using deuterated internal standards. And this is controls, and after treatment in the susceptible variety BML6, you can see there is not very high induction of JA and JA isoleucine, whereas in the CML71, which is a resistant variety, you can see a delayed induction. So after 6 hours, you can see JA getting upregulated and also JA isoleucine, whereas in DMRE63, which is a very highly resistant variety, there's a very high induction of JA and JA isoleucine. So this tells us that jasmonate definitely is a very good marker for screening, genotype and finding the basis of this resistance that we see in the two genotypes. Now, what was very surprising for us, that it was not only the levels of JA and JA isoleucine which was varying, but this form of JA, which is called a 12-OH JA. So 12-OH JA is actually an oxidation product of this plant hormone JA, and it's a metabolic shunt which diverts jasmonic acid from conversion to jasmonic acid isoleucine. So jasmonic acid isoleucine is the form of hormone which defends plants against herbivory, or it is the bioactive form. Now plants are also actively converting this jasmonic acid into an inactive form called 12-OH JA, because too much JA is also not good for the growth. So they have to maintain a balance of growth and defense, and hence they are converting it also into 12-OH JA. What we found during our quantification data was that in the susceptible variety BML6, the levels of 12-OH JA was highly enhanced, and resulting in the reduction of the constitutive level of JA. Yeah, that means the plant already has a very less constitutive pool of JA. Forget about herbivory. Under control condition, the plant doesn't have enough amount of JA to respond to herbivory, because it is getting converted very actively into 12-OH JA. So from all our phytohormone quantification data, we have figured out that higher JAOH and failure to induce jasmonates makes this variety BML6 susceptible to herbivory when you compare to the resistant genotype. You can see how high the 12-OH JA is in the susceptible variety, whereas in the resistant variety, they are very low. Now, there is not only hormones, but as I told you, secondary metabolites, which are also involved in maize response to any chewing insect pest. And one of them is this benzoxinoids, which are indole-derived plant defense metabolites in maize. You might have heard the name Dimboa. So this is Dimboa belongs to the benzoxinoid family of metabolites. So in plants, they are stored as Dimboa glucoside, and they are converted by a plant glucosidase into toxic Dimboa. Now, as young students or participants, you can always ask me that many of these metabolites are very harmful for the plants. So how do the plants store them once they make it? How do they store it that it's not damaging to them per se? So most of the times, plants, this is an example of benzoxinoid biosynthesis in maize. So benzoxinoid or many of the secondary metabolites are conjugated to sugars and stored as stable glucoside in the vacuole of the plant. So you can see this dash gluc. So these forms are not toxic in general to the plant as well. So they are stored as stable glucoside. When there is a mechanical damage by an insect or whatever reason, then there are plant enzymes called glucosidases. What are glucosidases? Anything which will cut the glucose out. So these act on this stable glucoside and remove the glucose, this moiety, and you get a toxic egg glucone. Toxic egg glucone is what is toxic to the insect or whatever is eating them. Okay, so this is the stable glucoside, is the stored form, and after mechanical damage, we get the toxic egg glucones. So in maize, we wanted to devise a method where we can quantify all of these metabolites because they are so critical for maize in defending against insect herbivory. So this is the whole pathway that, that results in the formation of these benzoxinoids. So we used a targeted LC-MS method for quantification of this Dimboa and different metabolites. After a lot of trial, this is a published method, but still there was a lot of trials and errors involved because we wanted to quantify all of them together. So we had to adjust their, various run conditions so that we have them as a high throughput method to quantify all of them together. So we have had a very good, person in our metabolomics facility, Ms. Khushbu Adlaka. She was initially from SciEx, then she continued in our institute, and she's the one who really standardized all of it, because in the end, it is, the people on the machine who are able to help us really well in this whole method. So she developed a method where she was able to quantify all the metabolites in a single run, at least with respect to maize. Now, once we had quantified, we had standardized this method, we measured all the forms, the gluc forms and the toxic glucone forms, everything we quantified together. This is in the control condition. This is the susceptible variety BML6, resistant variety. These two are resistant variety. This is under when it has not seen the insect, and this is after 24 hours of insect herbivory. You don't need my help to tell you that, upon herbivory in the resistant variety, you can see a very high induction of these benzoxinoid compounds. But one of these resistant variety, the DMRE63, also has constitutively higher level of this, metabolites per se. Now, not all of these metabolites are toxic to the insects, because over the time of evolution of plant insect herbivory, the fall armyworm has learned to detoxify many of the secondary metabolite Dimboa, but it cannot detoxify this H Dimboa glucoside form. It cannot detoxify the H2 Dimboa form. So these all we can quantify in this method, and you can see that the levels of H Dimboa, for example, is very high in our resistant variety in one of them, and in the other one, H Dimboa gluc is very high in this both the varieties. So from this analysis of benzoxinoid, we concluded that the DMRE63 is very high in benzoxinoid, both at constitutive, not only when herbivory is there, but also at constitutive levels. And the CML171, which is another resistant variety, has low benzoxinoid at the basal level, and it shows a fall armyworm induced biosynthesis of the benzoxinoid. Whereas your susceptible variety is susceptible because it is simply not able to induce any secondary metabolite, for in this case benzoxinoids. It is not able to induce at all. So this is one of the ways by which we have tried to identify how we can use metabolites as a markers for defense gene induction. So we not only did targeted, we had also done a lot of untargeted metabolomics in maize using Orbitrap. And we also have identified many other metabolites which are involved in the whole process. In nutshell, this is our very brief model. So this is the susceptible variety, the BML6. We have seen earlier that it has inactive form of JA, a lot of inactive form of JA, and it has low JA and total specialized metabolites which are important for herbivory. One interesting data which we got from our untargeted metabolomics was that this plant has a lot of metabolites which are induced, high metabolomic diversity. It's not that it doesn't induce anything. It is just that it doesn't induce those specialized metabolites which are important for herbivory. For example, jasmonate or benzoxenoid, it is not able to induce upon herbivory, which is resulting in a low defense. Whereas in CML71, it moderately induces JA and specialized metabolites. It has low metabolomic diversity, but high inducibility of metabolites which are important for imparting resistance to herbivory, which in turn results in an inducible moderate defense in these plants. The DMRE63, on the other hand, has high levels of all metabolites and hormones. It has low metabolomic diversity. However, it has very high jasmonate and benzoxenoid accumulation, which results in high defense in this genotype. So this is one of the models that we have derived from this work, a very recent work which we have been doing. I will, if you have doubts on it, I would prefer you can ask now or maybe in the end, because I'm going to the next topic where we have used metabolites. So in conclusion, resistance against FAW is regulated by, I haven't shown the primary metabolites, but you hear it from me that there are multiple primary and secondary metabolites involved in the process, and benzoxenoids, which are excellent secondary metabolites, which we can screen to, enhance maize resistance to FAW. And incidentally, it is known that all these benzoxenoid genes are located in a specific arm of chromosome 4 in maize, which makes it very ideal for marker-assisted selection if you want to, enhance resistance against this insect. So another very novel thing that we found is this jasmonic acid inactivation in the susceptible variety, which we are studying further. So this is with the herbivory part. I'll quickly, since I have some more time, I'll quickly go through the other topic which we are working on and how we have used metabolomics to identify the role in plant growth promotion. So this is a recently published work done by majorly by my postdoc Anish Kundu, and this in here we have used Piriformospora, which is a fungus which I told you, which enhances plant growth, and we have identified the metabolite involved in its growth promotion response in tomato. So as you know, P. indica interacts, colonizes the root of the plant and gives it growth promotion response, stress tolerance, everything. And we know that it enhances nutrient uptake, it results in phytohormone production. These are all known from earlier studies in Arabidopsis mainly. Yeah, so what we were interested in is, are they specific? We know about hormones, but we don't know of any other metabolites involved in this interaction in any major host with any functional role, which is known. So we wanted to know which functional plant metabolites are involved in P. indica mediated growth promotion, and we used tomato as a host for this fungus to look at this response and which metabolites are involved in this whole response. So we first had to standardize a system by which we will, at which time point to measure metabolites. These are the factors which we took into consideration. This is a tomato plant without this fungal colonization in the roots, and this is with P. indica colonization. You can see the plants are obviously very big when you cultivate it with P. indica. They colonize in the majorly in the roots, and they enhance both shoot and root fresh weight. So we decided based on this graph 30 and 40 DPI, these are the 3 time points that we tested. And since we got the major growth promotion at 40 DPI, a day post inoculation, we used this for our metabolomic analysis. So this is how the root looks like at 40 DPI, huge, big roots, from this fungal colonization. And you can also see, we also quantified how much fungus is growing on the plant, because why I'm emphasizing it more and more is at the time point that you choose also, determines what you get. So if you, for example, look at metabolites at early time point, you might be majorly getting those metabolites which are involved in this recognition process or early colonization in tomato. But if you go to a very later time point, you will also, be majorly looking at those metabolites which are involved in growth promotion response. So we have taken this time point. So we used in this case an untargeted GCMS analysis. This was done initially in AIRF in JNU before our metabolomics facility was established, at least this GCMS part. So what we did, we took the root and shoot of a tomato after co-cultivation with the fungus. And we also took the fungal mycelia because we also needed to know that the metabolites that we are identifying are really plant specific. Yeah, so we measured root, shoot, and my mycelial metabolome. And as you know, if you have done an untargeted GCMS, annotation is an issue many times. So this is what we were able to analyze, annotate from the mass signals that we got in leaf, root, and mycelia. Yeah. So from this, what we figured out that it alters obviously leaf and root metabolome, and we were, since it's GCMS, we mainly got sugars and amino acids, which were majorly, you know, at the first go, you will see that they were upregulated, but there were also many other specific metabolites. Now, when you look at untargeted data, it's like looking, putting in a needle in a haystack, because there's so much data in there that to find out something meaningful, you really have to analyze it very, very well to find anything meaningful. So in our case, our question was, which was that metabolite involved in growth promotion. So we took many approaches to reach there, and I'm trying to explain it to you in general. So what we looked at both the leaf and root metabolome, and what we found was there is a negative correlation between, at least for many metabolites, between leaf and root metabolomes. As you can see here, this set of metabolites are negatively co-regulated clusters. Since piri was majorly a root specific metabolome in endophyte, we decided to majorly look at those metabolites which are enhanced in the roots only. Yeah, and when we analyze the data from the root metabolome, we found that, at least from the annotated data we have, we found that this putrescine was one of the highest induced metabolite in our whole untargeted data. We also did this VIP score based method to determine the putrescine, importance of putrescine in the whole data that we got. So once we figured out that putrescine may be critical, we also took out, so this, though putrescine was one, we also tested all the metabolites which were in the second and third position here, or which was highly downregulated. All of them also we tested, but I'm just showing you the putrescine data. So what I showed before was a GCMS untargeted quantification. If we had to do any study which will further, we had to devise a method by which we can accurately quantify putrescine in plants. So we used LCMS in this case. This was done in our facility. We develop a method by which we can quantify putrescine using LCMS, accurate quantification using a proper standard. And this is the level of putrescine in the roots. The mycelia also produces a very negligible amount of putrescine, but it was mainly plant derived in our case. So now when we quantify this putrescine, you can see upon fungal inoculation, the levels of putrescine are highly increased in tomato. Once we knew about that, we looked up at literature. What is this putrescine? Is anything known about it in any other interaction? And we found that in rice, it's a common signature upon interaction with plant growth promoting rhizobacteria, where they are showing increased accumulation of coumaryl putrescine and but these are all just reports. These are just reports which tell me that, okay, putrescine is induced, but there is no functional role here. Nobody has knocked out a gene which is responsible for putrescine and seen an effect on any of the system. Yeah, that is what, as students, when you look at metabolite data, you will, this is what I want to tell you, you should always look at what pathway is responsible for this, and if you knock out the pathway, what is the impact on your system. So this was what we tried to characterize in tomato and with P indica interaction. So putrescine is a polyamine. Actually, it occurs in free form, or it can be conjugated. It can occur in conjugated forms, and these are putrescine, spermine, spermidine. These are abundantly available in plants. Yeah, they are known to be involved in plant growth and development in general. So what we did, we wanted to first to ask why does plant induce this putrescine level of upon colonization? Does it in any way benefit the fungi? Because if there is no benefit during an interaction, it would be strange for it to induce it. So we looked whether it is beneficial for P. indica in general, and we found that yes, it is beneficial for P. indica up to a concentration. So it is very concentrated. If you put a lot of it, it doesn't become beneficial. So you can see at 10 micromolar, the P. indica radial growth increased, but it saturates after that. So if you if you treat the P. indica fresh biomass, you could get increased biomass if you treat it at this specific concentration. Yeah, this is what you see here also. So yes, this metabolite is beneficial for the fungus as well. We'll go to the plant in the next slide. So when we look back, we found that putrescine and spermidine are involved in regulating hyphal growth in arbuscular mycorrhiza, because this is a growth limiting factor for in spores which is found. So there are reports of this putrescine in earlier systems, though no functional role was known. Now we asked ourselves what advantage does it do to the plant? Easy experiment. So we tried to add putrescine externally and look if plant growth is enhanced. We got a enhancement in shoot and root growth when you treat putrescine, as you can also see in seedlings. However, this is not enough to tell me whether putrescine has any real role in plant by interaction with P. indica. So we also measured what happens if you add this putrescine. Does it also affect any plant hormone pathway? Why we looked up, because P. indica is known to activate auxin production majorly when it interacts with plants. In various plants, we have shown this. This is the picture of, auxin reporter and P. indica colonization for different times. And you can see that the auxin is upregulated when they are interacting with each other. So we asked ourselves, does putrescine in any way, regulate this auxin biosynthesis? So for that, we had to measure auxin, and this is in control and putrescine treated plants, and we could get that putrescine treatment induces auxin. And then other growth hormone that we checked was GA and cytokinins. So in cytokinin, we didn't get any major upregulation of cytokinin upon putrescine treatment, but we found that GA4 and GA7 were upregulated when you treated with putrescine. So again, this was done by the LCMS based quantification that I described to you in detail before. And it also shows the capability that we are able to, quantify all of this accurately. So from this data, we can tell us that putrescine increases growth promotion in tomato by elevating IA and GA, but not a cytokine. Now we went into the genes which are involved in these processes. What will happen if the specific gene is knocked out? So putrescine is produced either through from ornithine or arginine as precursor via arginine decarboxylase or ornithine decarboxylase. We wanted to know which of these are involved in our system, and so we did a real-time PCR to figure out which one of them were induced when this fungus is colonizing the roots. And we found only one member which was highly upregulated, which was the SLADC1 or arginine decarboxylase 1. So they seem to have a root expression. It is published, they are majorly expressed in roots. And then we decided to look what will happen if you don't have this SLADC1, if you knock it out, what will happen. So for this, we devised a method. We used a Wix when we silence this SLADC1 in tomato, and then we grew these Wix plants along with P. indica, and then we looked up what happens to the growth promotion. So for any Wix experiment, which runs for a long term, because this is a co-cultivation which goes on for 40 days. So Wix is a transient method of gene knockout. So we had to make sure that our SLADC1 is down-regulated at all the time points that we are tested, which was the case. And we also measured the putrescine levels in our Wix line. So we were sure that our Wix line has reduced putrescine levels. And then we proceeded with this experiment where we co-cultivated the roots with P. indica. So this is what happens when P. indica wild type in wild type, you can see a very nice growth promotion. This is with the empty vector. This is with the SLADC1 VIX. So you can see in SLADC1 VIX plant, there is absolutely no growth promotion. If you look at the quantification data as well, you can find it wild type plus PI. There are some outliers as well, but they are statistically significant. This is empty vector plus PI, and this is when you knock out the putrescine biosynthetic gene. So there is absolutely no growth promotion both in the shoot and the root with the root fresh weight. And we also found that there are auxin response elements which are found in the promoters of SLADC1, which could explain its auxin effect. So once we had this data, this SLADC1 is the gene responsible for the putrescine biosynthesis in the system, and it's critical for this interaction. We also we have seen that it doesn't promote growth. So as a reviewer of a paper, you can ask me, yeah, maybe it doesn't promote growth, but why? Is it because the fungus is not able to colonize it, or is it because there is no putrescine, or does putrescine have any defense phenotype? So to know that, we quantified the levels of fungal DNA in wild type plus PI, in empty vector plus PI, and SLADC VIX plus PI. So from here it is very clear that in the absence of putrescine, the fungus is not able to grow on the roots, or it is this metabolite is important for fungal colonization per se. Yeah, so as you can see, very reduced colonization at 40 and 30 DPI. Yeah, so you have already seen how this metabolite is crucial for the P indica per se. So this might be the reason why it is when it is not there, the colonization is less. This is what you see also with the imaging, where this is in the empty vector and this is in the SLADC1 VIX plant. Now the question was, is this pathway conserved in other hosts? Is it only in tomato that you see it, also, or also in Arabidopsis, which is another host of P. indica? Since our lab is an Arabidopsis lab, it was not very difficult to do this. So we did it in, we treated also putrescine on Arabidopsis, the specific concentration, which I tell you, which is important. And we see a fresh weight increase. Then we ordered lines in which the SLADC or the corresponding gene in Arabidopsis, the ADC, are all knocked out. So we ordered multiple knockout mutants in Arabidopsis to check the functional role if this gene is not there. So this is the control, and in Arabidopsis, the PI treatment, you can see growth promotion here, and this is if you complement with putrescine, you cannot see more than that if you complement. Now, if you, in this ADC mutants or those plants in which the ADC genes are knockout, you can see upon P. indica treatment, there is no growth promotion. However, if you supplement with putrescine, you are able to get back the phenotype, which tells us that this is what we quantified here as well for multiple lines. That putrescine is responsible for P. indica mediated growth promotion, and it is rescued by exogenous application of the putrescine. So we also did similar experiments on colonization in Arabidopsis as well in this mutant, and as you can see, they are not colonizing so well on the ADC mutants. So telling us that this putrescine is important for P. indica mediated growth promotion functionally as well. This led us to this model whereby this is the colonized P. indica which grows in the root cortex, which this interaction between roots and the fungus results in a metabolomic rearrangement, and we get arginine mediated production of putrescine is enhanced. This increased putrescine in the plants results in increased IAA and JA, which is responsible for plant growth promotion. Yeah. Now, this is not only important for the plant, the putrescine production is also important for P. indica growth and colonization per se, and hence it is, a both way beneficial in this whole process. So it is co-adapted by both host and microbes. Yeah, so with this, I'll just come to the end of my talk and also tell that our facility has been responsible for most of the measurements that I'm showing here now. Was inaugurated in 2019, and we are able to quantify plant hormones, amino acids, flavonoids, many majorly plant metabolites that we are able to quantify. So we are not specialist in lipids, for example, which Ujjaini is doing so well here there. But our main thing is the phytohormones. So these are the equipments which we have, and please, any of you, if you have any samples you would like to, ask similar questions in your study system, please get in touch with us. Yeah. So I would like to thank everybody who is involved in this work, my whole group. This is our institute in near JNU campus, and we have multiple collaborators and funded by different sources. So thank you very much. I'd be happy to take questions.
Thank you, Jyoti. It was particularly very striking to me that using the same tool during the first story, you showed how the plant growth was retarded, and in the second story, you showed how the plant, grew better.
Yeah.
So with that, I think we'll start taking questions. I will just take the mic. I hope you have a little bit of time.
Yes. I'm home.
It was an excellent talk. Thank you for this. Actually, my question is that in your first part, you had estimated the J pathway, but after J, you have seen some another antioxidant enzymatic assay which was involved, like SOD, catalase, and another physiological parameters which affect your growth also. In you also after that, in the second part, in my second question is that in tomato, why you are not exceeding your experiment only for developmental stage, not taking away from the all over the field? What will the effect of on yield when we treat it is with or without that particular fungus? Thank you so much.
Yeah, thank you for that question. So I'll start with the first part. So upon herbivory, the major objective of this whole study was to screen germplasm and look for those resistant genotypes, and what are those metabolites that I can use in future to screen a whole germplasm. So using metabolomics. Okay, so when that is the question, we looked for those things which are absolutely critical. Of course, peroxidase, catalase, there are so many things, which enzymes, which are proteinase inhibitors, amino acids, hundreds of things are involved in herbivory. However, jasmonates and benzoxinoids, at least in the case of maize, are the most dominant, metabolites which are important in the pathway, and where a functional role is worked out. What is functional role? I have knocked out the gene for that jasmonate biosynthesis, or somebody has done that, and they have found that if you knock out this jasmonate biosynthesis, then this whole interaction is affected. So we are looking only at those metabolites initially for which a functional role is known. Peroxidase, catalase, they all come, but there is no functional role known per se in herbivory response. So for these studies, we have only looked at jasmonate and benzoxinoid, for which a functional role in herbivory is well known. So for the second question, I think you're asking whether it affects the yield. So we haven't done the yield part at all with the tomato, how it impacts the fruit growth. So this is something we can do, but we haven't done. We've only limited ourselves to this vegetative growth, because you've also to think that how do we do a functional assay in these plants? We are using Wix, which is a transient assay. So we cannot look at the fruit development, because that Wix will not be active for so long time till fruit. So if we will have to do a CRISPR line, or we will have to establish a CRISPR method or a knockout method, or stable transgenics we will have to generate if we have to go for fruits. So this is something that can be tested, and this would be also good. I'm sure there will be some other metabolites in fruit development, for example. So this is something that can be done.
Thank you for the talk, ma'am. I have a very basic question. Here we are trying to resist our plants to the insects. My question is, in the food chain, when we eat those fruits, do that also affect us?
I didn't hear you clearly.
Okay. Ma'am, the fruits that raised from these plants, if these fruits are eaten by humans, does they also affect us? Because we are modifying the plants to resist from the insects. And the similar things happen when we use pesticides.
We didn't modify anything. So we are only trying to look under a natural condition. Why is tomato growing bigger when you have this fungus? So we are only trying to understand why does tomato grow bigger when Piriformospora indica is inoculated on the roots. We just want to know which are the genes involved in this process. We only try to knock out that gene and try to ask ourselves, if this gene is not there, does it promote growth? That's all. We are not doing any, genetic manipulation up to now. But yeah, in future, maybe, we can look at targets which enhance growth promotion. But here in this study, we have just used, found out the pathway which is responsible for putrescine production, the metabolite that is naturally there in tomato when the fungus or interacts with the roots. Okay, so does that answer your question? Is this what you asked?
Hello, ma'am, can you hear me?
Yes, I can.
Ma'am, it is about a personal experience at my home when I was growing this tomato plant at my home. So I was desperately waiting when the fruit will come and I will eat it in the morning. So because you see, everywhere there are pesticides and everything. So I wanted a fresh fruit like I usually had in my childhood. So what happened is after a month or so, there was green tomatoes. They appeared on the plant. So I thought maybe in next 15 days I'll get the fruit. But what happened suddenly is the plant, died out of, I don't know what was the reason. So it got, so I was disappointed. So I'm tempted to ask that, can we use this fungus? Because all of the experiments you did was in controlled environment in your lab. So if we use this fungus outside, at homes, is this possible? Uncontrolled environments and everything.
Yes. So this is a fungus which is isolated from natural condition. It was isolated from the Thar Desert many years ago by Professor Ajit Verma. So it can be used. In fact, there's also a formulation of this fungus, if I'm not wrong. So you can use. But that's about tomato in your garden now. But as a student, you are sitting here, you tell me you've done this course for many days. You told me that your tomato fruit doesn't grow bigger. So what is it that you will do? Using your knowledge from this whole course, what will you do to identify why your tomato doesn't grow, or whatever, if the fruit is green or whatever? So how will you use this knowledge that you gained in this course for
Yeah, right. Yeah.
What will you analyze if from this talk? You have heard this talk for so long. Tell me,
yes.
what will you now analyze in this tomato fruit if you were given all this fancy mass spec? So what would you analyze now?
Ma'am, you see, I observed that plant died suddenly. Like I woke up in the morning and I saw that leaves are all gone and everything. So I think there was something different, maybe not an insect, maybe some other reason or something like that.
Yeah.
Yeah.
So you can easily quantify. So if you were given all these methods, you can use as I'm talking about phytohormones being markers, you know, I can use this as a marker and try to figure out how does the hormone change. For example, in your dead plant, is it, if it's due to an pathogen, it will be mainly salicylic acid which is induced. If it is due to an insect bite, it would be jasmonic acid which is upregulated. Apart, of course, so you can use your knowledge that you gain from here in future. This is what the you know, essence of this whole thing is.
Right. So again, the next question is, I have time, right? Yeah. So I was like, it was in extension of the previous question. Was it was just a growth you measured, like it was faster, or you did measure also the quality of the leaf, or for example, surface area and everything? The leaves are rigid or not?
No, we just measure the shoot and the root fresh weight.
Okay. Thank you so much, ma'am.
Thank you very much.
Excellent talk.
Hello, ma'am. Thank you for that amazing talk.
Thank you.
In the first part of the PPT, you included the jasmonate signaling, and in the indirect method, there was this volatile signals. I was curious that how do you study the volatile compounds in the plants? Like how do you quantify it?
Yeah, thank you very much for that question. So volatiles, it's a whole field on itself, volatilomics actually. So you can measure, trap these volatiles. We use special trap, SPE filter trap, to trap these volatiles, and we measure it using GCMS. So you, for example, you can also trap volatiles in the field. There are also methods by which you can take these filters into the field. You can cover each of your plant and trap your volatiles in the special filter. You bring it to the lab, and it is usually analyzed by GCMS. So it is, if you're interested, please write to me. I can also send you the details, both in lab and in the field. Essentially, it's GCMS based measurements.
Ma'am, first you had to know that there was something which was like volatile there, that you had knowledge from previous pathways, or like?
No, for volatiles, see, for many plants, it's nobody would have any knowledge. I think if you trap via this method, volatiles is what is released out, right? So we have a very good volatile library that is available as well for annotation of these volatiles. So you can do a lot of untargeted, mainly untargeted is done in volatile business. So you can get a feel of what volatiles does this plant induce in general before you go further. But everything, this is GC based method. There's also a portable a volatile machine called a Z-nose, which you can carry to the field for people who are doing field work. They have this Z-nose that they carry to the field as well, essentially using these filters, and they'll come back and do GCMS. That is it.
Okay, thank you so much.
So in maize also there are many volatiles. So we are ourselves not have not established the volatile part very well, but this is something we want to do in future. Yeah.
Good afternoon, ma'am. I am Dr. Pawan from Himachal. And then one basic question: can hybridization reduce the less use of very less use of these pesticides and less dependence on environmental factors like water? Like maize, it depends on the water mainly. So hybridization.
What did you ask? Hybridization?
Yes. Intermixing of species and some special species.
In maize, usually we grow hybrids, yeah, at least in the case of maize. So, however, we have to select for good inbreds to make that hybrid. So, in our screening method that we have used, all these varieties, they are maize inbreds. And if you have seen the picture, the inbreds which have higher defense have compromised growth. So the whole idea is they will cross it with a very high yielding maize variety, because in maize we are always cultivating hybrids. They will cross this resistant variety to a very high yielding variety and try to, introduce these genes which are involved in the major secondary metabolite pathway, so that it doesn't affect the growth per se.
Right.
Did I understand you correct, or was it something else?
No. Thank you. Very good afternoon. It's really very fascinating work you have done.
Yes, thank you.
The mechanism of understanding of how plant defends and how they induce the growth promotion. So when it comes to growth promotion, I think my other two friends asked yield. Yield is also very important, and also we should see the nutritional values in the food is what's matter, because that's the end thing which we as a scientist think, and it's good for humanity. So I think those parts, if you do, I think that it's a good work.
Very good suggestion. We have not done it, but yeah, nutrition is something I would be really. I just thought when you asked me, so it is a really nice point. We would definitely be checking if we go to the fruits. That is, yeah.
Thank you.
Yeah, but I'm sure there are, I'm this one metabolite that I told you is at a very specific stage. Maybe at a later stage, fruit development, there might be something else which is, playing a crucial role. So this is all, ongoing. It can go on for years, how we study this whole interaction.
Because recently I saw, many scientists say, there is so much yield, higher yield is there. Everybody is thinking about the higher yield, but what about the nutritional value?
Yes.
That nobody is thinking.
It is not very difficult to measure the nutritional value. So for example, you might have also learned in your course as well, we use the ionome profiling using ICP-MS to quantify what are the, what are those nutritional and enrichments that is happening. So it is not very difficult to measure. We know with P indica that it enhances phosphate, nitrate uptake, but nothing more. So this is really an open field.
Hello, Jyoti, I have a question,
Yes.
and they are all very naive questions. So the first one is that in your first story, I also did notice that your resistant maize was little short in stature, and growth-wise also it was a little smaller.
Yes.
Is there a first of all, what I wanted to know was what was the molecular basis of it? Did you work on that? That why is it what happened in these plants? Which were the genes that were that were deregulated, or there was differential expression? Because not always gene deregulation is correlated to metabolome change. So could you figure that out also, that what are the molecular changes happening, and whether those have what kind of correlation they have with the stature of the plant, with the structure of the plant, that a little short stature of these resistant varieties.
Yeah. So thank you for that question. It's very true that the resistant variety, at least the DMRE63, is a really small plant. So what we know, we have also looked up at the genes which are involved in the whole pathway, which is upregulated upon herbivory, and we find that the benzoxenoid biosynthetic gene cluster, the 1 to 14, there are 14 genes in this whole pathway. They are highly induced in this resistant variety. As I told you, they are all sitting in one place in the chromosome 4 of maize. So at least transcriptionally, they are also behaving like the metabolite data. They are also induced. And what you tell is correct. Many times the metabolite induction pattern and the gene expression patterns do not match. At least in Arabidopsis, for example, glucosinolates are the marker for defense. There, though the glucosinolates are induced, the corresponding genes are not, they don't really align with each other. But in maize, they do. So the benzoxinoid genes are found to be higher in the resistant variety compared to susceptible. Now, regard to the growth, for the DMRE63, our current hypothesis is because it has constitutive or in control condition itself, the level of secondary metabolite is so high in this plant. This must be the reason of this, decreased growth, because at some stage it is also costly for the plant to keep on producing this secondary metabolite. So this can be, but this is something we are working on. The good thing is what I've heard from the maize breeders is most of the resistant lines usually have a yield phenotype. I mean, they look smaller and they yield lesser as well. But in maize, since hybrids are cultivated, these are just inbreds, or these are just the parents that will be used to make a hybrid. So they will essentially cross it to their high yielding varieties, and this will be taken care of when they select for hybrids. That is what they are telling. They will, when this hybrid production is done, this low yielding thing will be taken care of. But the varieties that we have tested are their parents.
So just one last question, so when you say that the benzoxinoids are high, is it some kind of a dominant negative mutation, or is it some kind of what happens at the molecular level?
No, we haven't worked that out, why it is high. From the literature, for example, there are some reports in certain lines that they are majorly, for example, there is some mutation in the benzoxinoid 14 gene, which results in higher upregulation of benzoxinoid in certain lines, but we haven't reached there, whereby we know why it is high in the control conditions.
And at the very end, so is it only Solanaceae family that grows so well with P. indica, or others also?
Everything. There, I haven't found a known host for P. indica. You take any plant, I think it
but
grows.
That's a big prize, big boon.
Yeah, it is. And it is a non-transgenic approach to enhance plant growth. I mean, I don't know why it has not it has not grown. Its use in India has not increased, but there is a negative effect. At least we try to look at whether, P. indica gives it resistance against herbivory, because my lab works on insects. So we try to ask ourselves, if we make this fungus grow on the root and the plant becomes big, so does it in any way give resistance against the insect? And the answer is no. So it actually makes the plant more susceptible to the insect. So it gives resistance only to certain classes of kinds of pest or microbes. So there is a cost associated with this growth promotion as well. This is something we have to figure out.
So you need to tamper the benzoxinoids together with P. indica.
Yeah. So maize we have not done. P. indica maize is too much, I think.
Okay, so thank you so much. And can I give your email ID to the participants?
Yes, I would be happy to. I feel so better also after giving the talk. My energy.
That's good. That's good. At least you are smiling. In the beginning, you were.
I was like dead. Yeah,
no, but it was really good, and I'm sure all of us enjoyed. And next time, I would really want you to be with us here.
Sure,
so with that, then we'll end this session. Thank you. Thank you so much.
Thank you.